In this video, we will continue our discussion about polysome profiling. In the previous video, we have discussed different methods that are initially been used to compute translatome. This translatome means the mRNA that are being translated within the samples. So let us start with the material preparation. So in this material preparation, we need to first prepare the polysomes. So one of the strategies is to extract cells from the sample, or from, in this case, from the animals, and then lysate the cells and filter out the nuclei or cell debris, and take the cytoplasmic lysate containing polysome into the different layer of sucrose gradient that we have discussed, from 10 to 50% gradient, and then ultracentrifuge it and uh, fractionate them out and measure using the profile to isolate the free RNA or free RNA, such as the free ribosomal part to 20S, 40S, 60S, 80S RNAs, and uh, separate out them from the samples, and only the polysome will then be isolate and quantify using different strategies, such as RNA-seq data that is shown here. And another approach is that we can take the tissue of the whole tissue of the animals, and then pulverize the tissue and lysate the cells within the tissue, and follow the same process, that is to extract cytoplasmic lysate, and then extract polysome from this cytoplasmic lysate using sucrose gradient, and quantify to analyze the translated mRNA, or mRNA that are being translated within the cells. So in this video, we will also cover in brief about how this RNA-seq data, how this quantification works. So these are the RNA-seq data that we obtain from the samples, polysome samples, and after that there is a transcript abundance, or we can say transcript abundance is measured from different fraction, as the samples that are obtained, or the polysome samples that are obtained are from various fractions of this sucrose gradient, and later on they will compute with respect to monosome in order to identify which transcript has ribosome bind to it, and then they will plot this graph, that is ribosomal occupancy description, and comparing it with control. As we can see that this is the control experiment, and this is the actual experiment. That we can see that there is a decrease in the shift of ribosomal occupancy, and later on this output can be is generated in forms of table, and one of the table example is shown here. uh, Let us go through one of the rows. That this is the transcript, and the second column is shown the gene name, and third one shows its RRO value, or we can say relative ribosomal occupancy within the control. And next, the this column D will shows the error. What is the error based on the different number of experiments, different number of fractions that are been analyzed. Later on, this RRO from the experiment they identify, and its error value that is shown here in the column number F. And then in order to account for the in order to account for its significance, this p value has been calculated through this test, and then the false discovery rate has been calculated through this q value. Later on, this ROS is calculated. ROS is just a ribosomal occupancy shift, whether the ribosome is bind to this gene or not, and comparing it with the control uh, uh, control value, they can identify uh, whether there is a increase or decrease in the ribosomal occupancy. In this case, since it's the negative value, there is a decrease of the ribosomal occupancy shift. So there is a decrease in the translation of this gene X. Similarly, for this next gene, gene Y, this value is almost close to zero. So there is almost no change in the translation of this gene Y. Whereas for gene J, it is a value is positive. So the G value is positive. This indicates that the more ribosome is bind to this gene Y in the experiment case compared to the control. There is an increase in the mRNA translation. This value is not compared with any of this column that I have said earlier. I want to correct that. These values are calculated based on this Sirlic output. There is a algorithm that is when present within this Sirlic analysis. And if the values are positive, very high, greater than zero, then it is considered as there is increase in the mRNA translation. If the value is too low, such as this, in this case -15, then it is considered as there is a down regulation of the mRNA translation. If value is close to zero, then there is no change in the mRNA translation. And this is the visualization how it is, how it can be observed. In case of increased occupancy, the ROS value will increase, and this shows that ribosome binds to the gene more compared to the controlled experiment. In this case, there is no change, and in this case, there is a decrease in occupancy. And this thing can be visualized using ribosome occupancy shift using this uh, density plot. If it is close to zero, there is no change, and if it is in the negative side, there is decreased ribosomal occupancy. If it is in right side, there is increase in the ribosomal occupancy, or RRO, which is the relative ribosomal occupancy. And this thing can further be analyzed using the heat map that is being shown here. Here the different comparisons are being made. First, within the different fraction of the control, the fraction 1 and fraction 2 of the control, and fraction 1 and fraction 3 of the control. And then the comparison are also made within the experiment of different fractions. And these fractions are obtained from sucrose gradient. We can see that in the fraction 1 and fraction 2, there is some changes, and in fraction 1 versus fraction 3, there is some changes. This shows that what are the ribosomal occupancy changes compared to the first fraction. So in this way, the polysome profiling has been carried out, and this is not this does not cover the whole in-depth understanding of this polysome profiling. If you need to understand this polysome profiling in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.